Thank you everyone for joining. We're here to talk about what we've been up to at Portal and how some of our potential partnering could be helpful to others out there. Just for background on Portal, we started early in 2023 and we're based on next-gen mechanical delivery technology. At this point in time, the company has had over 13 million in funding. We're about 14 people and we have over 50 active partnerships right now across biotech, pharma, academia, including more than half of the top 10 pharma. So there's been a lot of different activity given what the technology can do, and we're happy to talk through what the core platform is and how we can think about applying it across drug discovery applications, cell therapies, and other areas. So the main problem that we're trying to solve for is how do you put materials into cells? Because ultimately cells are the little biological machines that make everything happen in our body. And being able to put the material into cells is important both from a drug discovery perspective and from the perspective of cell therapies, where you're going to have to engineer a different cell function. If you think about most of the techniques that people have available right now, especially for cell therapies, there's a few different categories, for example, viruses, lipid nanoparticles, electroporation, and all of these systems have their benefits, but they also have significant challenges where they struggle to get integrated with other systems, or it's difficult to preserve cell health. There could be scaling issues. There's a lot of restrictions on cell type and cargo, and delivering multiple things at once is not trivial. Whereas with Portal, we feel like we can actually help solve a lot of these underlying issues and eventually have a system that can put virtually anything you want into any cell type and therefore engineer many different functions that aren't possible so far. The core of how our technology works is that we take cells, we mix it with the cargo that we're trying to deliver, and then we run it through what looks like a fancy filter. So it's a thin piece of silicon with holes in it. These holes are designed to be slightly smaller than the cell, so that as the cell goes through, it gets rapidly deformed or squeezed. That ends up disrupting the outer cell membrane such that anything that's outside can then diffuse into the cell for the about 30-second window where the cell is open. So it's a very simple, straightforward way to put virtually anything you want into any cell type that you want. Now, if you think about what the delivery field looks like today without Portal, and we had a review in Nature that's cited at the bottom from 2016, but this is kind of an oversimplified summary of it. You could think through the matrix of what are the cell types that we're trying to work with, and then what are the cargoes that we're trying to put into cells. There are certain things right now that are relatively easy to do. So for example, putting nucleic acids in cell lines is not that difficult. Putting in a virus or lipid nanoparticle or electroporation can often accomplish that. Then when you get to primary cells like immune cells or stem cells, it starts to get tricky. There are certain things you can do, there are certain things you can't do. Cell function tends to start to become a problem, hence why that's a bit of a yellow zone. And then finally, there's a lot of material classes that are virtually impossible to deliver reliably, such as proteins, peptides, impermeable small molecules, et cetera. And that's because most delivery technologies really rely on the high negative charge and chemical uniformity of DNA and RNA for them to work. Whereas when you get more diverse, when it comes to small molecules and peptides and proteins that can have a really large range of chemistry and charge properties, this all falls apart. It becomes very difficult to deliver them. And with Portal, we completely changed that because with our mechanism, it's just diffusion-based delivery. So it doesn't really matter what the material is. We just enable its diffusion into the cytoplasm regardless of chemistry or charge. And so we completely changed the matrix of what you can and can't do, especially because we turn a lot of areas green that were previously yellow or red. Now, the one weak point for us, though, admittedly, is that we don't directly enable nuclear access, meaning that if you have a piece of DNA, we would just deliver it into the cytoplasm. That piece of DNA is going to have its own way of getting into the nucleus, and that's why this is a yellow zone. For certain cargoes like CRISPR RNPs, that's not really an issue because they have nuclear localization sequences on them, and so even if you put them in the cytoplasm, they will get chaperoned into the nucleus on their own so they can do their editing. Beyond this flexibility of cell type and cargo, the other big advantage for us has been we keep cell function normal. So across our publications over the years, we've shown that with mechanical delivery, for example, T cells will proliferate normally after they've been mechanically delivered, whereas with things like electroporation, they can be deficient. Or hematopoietic stem cells will engraft normally with mechanical delivery, whereas with electroporation, again, they tend to be deficient in their engraftment potential. And this all comes down to gene expression. So if you're looking at the gene expression profile over here, these are from data where we did a full transcriptome microarray of hematopoietic stem cells that had been, in this case, electroporated, and then compared it to untreated cells, where black means nothing changed in gene expression, red or green means there was significant up or down regulation. And you can see that electroporation lights it up like a Christmas tree, mechanical delivery keeps things pretty normal, and that is what illustrates why we can keep cell function pretty normal while having this diversity of putting virtually any material into any cell type. For implementing our technology, there's 3 different scales that we've been implementing it at. There's the research scale system, which you see on the far left. There are high throughput integrations for doing high throughput screening or other activities that are plate-based. And then there's the clinical scale systems where you can do billions of cells for clinical use cases. And one important thing to note as far as how we implement Portal is that for this high throughput and clinical scale, we actually are just a cartridge that connects with existing machines. So we're not trying to make a bunch of different new capital equipment, but rather leverage existing infrastructure and or get integrated into new equipment that people are creating. For our basic research scale system, the core of this is the gateway. So the gateway is this unit you see on the left. The base of this is about the size of a tissue box. And the cartridges you see on the right, these are the consumable components that contain the silicon membrane within them. And that is what disrupts the cells to allow for delivery of the various materials. And one thing we can do is also help show a quick video of what this actually looks like in practice. And so in this case, we're doing a mock run. If you, sorry. If you assume that the buffer contains the cells and the cargo, we just pipette it into the top of the cartridge. That cartridge has the silicon membrane in the middle of it. So that's currently holding the column of liquid above it. And you set the pressure on the gateway, which is what determines the speed at which the cells are going to go through. Once you've set the pressure, we're just putting in a little Eppendorf tube that's going to be the receiving vessel for it. And the cartridge just lure locks onto the top of the gateway. And now once everything is in position, when you press the button, it goes through within about a second. So it's all a pretty fast process. And now you're done. The cells are open, material is diffusing in. You may not want to dilute it for the next 30 seconds, but other than that, it's pretty simple to do one of these deliveries at the research scale. So going back to the main presentation of this, we went through the process a little bit over the video, but one of the key pieces of data for us has been starting to look at primary cells. So for example, with T cells, we've shown that we can deliver mRNA and get high efficiencies while maintaining decent viability. So in this case, you're looking at over 90% delivery efficiency and close to 80% viability. We can also deliver multiple things at once. So in this experiment, we're showing the delivery of mRNA plus a CRISPR complex plus a dextran sugar polymer, and your triple positive rate is just over 85%, meaning that 85% of the cells contain all 3 cargos. And if you're looking at the FACS plots to the bottom left, you can see that virtually all of the cells that are expressing GFP are also knocked out for the B2M. And we can also apply this not only to mixed cargos, but also mixed cell populations, where in this case, we're showing delivery to mixed PBMCs, where we take PBMCs, we load them with mRNA, and we show that all the different subsets are capable of expressing the RNA, and you can again maintain decent viability throughout this process. Another flavor of looking at this is sequential deliveries, where in this case, we were taking unstimulated T cells, adding TRAC RNP, and then activating them, and then taking those activated cells a few days later, delivering mRNA and a B2M RNP this time. And now when you do this sequential delivery of multiple materials, we get about 50% of cells that are triple engineered, meaning they are double knockouts and they are expressing the GFP. When you do sequential deliveries, I would think of them as independent events, meaning that if the first event was 70% efficient and the second event was 70% efficient, you're going to get 49% of cells where both events occurred. We've been doing quite a bit of work with stem cells as well. So this is looking at iPSC expression for mRNA, as well as they're maintaining pretty good viability. And we've taken this a step further to more functional use cases, where, for example, in this case, you're looking at, we took T cells and delivered circular RNA for a CD19 CAR and a membrane-bound IL-2. So it was simultaneous delivery of two different circular RNAs, and we're showing that you can get really high killing effects with these double-engineered CARs, because for us, again, it's trivial to put in multiple things at once. So we can not only put in the CAR, but also put in something like IL-2 that will significantly enhance their activity. In a related space, we've also shown that with iPSCs, we can deliver transcription factors. In this case, this is an mRNA for NGN2, which is an important transcription factor that drives towards a neuronal cell fate. And you can see that we get good expression of the NGN2 itself, but also upregulation of the downstream genes that lead to a neuronal phenotype. Switching gears towards high throughput. The way we think about doing high throughput is that we want to implement it in tandem with existing equipment and workflows. So one example of one of our implementations for one of our big pharma partners is over here. What you're looking at is this robot is one of those liquid handlers that typically is just dispensing into one well at a time. And we added in a cartridge that sits in between that contains one of the portal membranes. And the way this works is that as cells go through, they will go through the membrane, get mechanically disrupted, and then you can actually just have the cargo in the well, because once you disrupt the cells, their membrane stays open for about 30 seconds. And if you have the cargo in the well, they will still take it up. So it's a simple way to essentially have a porated cell dispenser where you can have different cargoes in the plate, and they will just take up whatever's in the plate. And we've shown that you can get pretty consistent delivery. So you can do 5 to 10 plates per cartridge with this and maintain relatively reproducible delivery throughout the plates and throughout the wells as you go through it. One of the areas of application that more in the drug discovery world, people have been really excited, and especially is related to high throughput screening, is if you think about the current way in which people investigate impermeable molecules that they find, is that they had a biochemical screen where they found some molecules that bind their target but have been impermeable. And in the current dynamic, people will then go and do a significant amount of medchem optimization to make permeable versions of these molecules before they can put them in a cell-based screen and then see if it actually works or not. So a lot of times it doesn't work and you wasted your time and money doing this medchem optimization. With Portal, one thing that we've had a lot of traction on is you can actually take the molecules you identified in the biochemical screen and deliver them directly into your target cells. And by doing that, you get to immediately see, would these molecules have worked? If they did work, then you can go do the medchem optimization, and it's well worth the time and money to do so. Whereas those that weren't going to work anyway, you never wasted your time pursuing them. And we've already had publications in the past, for example, with Pfizer, where we showed these principles that you could take impermeable small molecules, deliver them into a cell, and understand their function better before you had to go and do medchem optimization. And in these papers, we were also showing that methods like electroporation were incapable of yielding the same results. So they couldn't be reliably used to deliver these impermeable molecules. Beyond that, we've also been exploring use cases where we can deliver materials like DNA-encoded libraries, because with DNA-encoded libraries, even though the small molecule component may or may not be permeable to a cell, the DNA tag definitely is not permeable. And so in this case, we've been able to show that we can get robust delivery of these DNA-encoded libraries into primary cells and cell lines, which could enable a lot of new assaying capabilities as people increasingly use DELs to understand different target spaces. Another illustration of our use cases in the drug screening area has been, in this case, showing the delivery of multiple cargoes simultaneously, such as antibodies and labeled oligos, or labeled polymers that can be used to investigate and query different aspects of cell function or different aspects of molecular activity. And you can see that in these data, all three of these cargoes go in at once, even though they are, again, chemically very different, and we maintain high efficiencies in these assays. Another zone that's on the flip side of this that has been exciting for us is delivery of probes into cells to try to measure different things within a cell that would have previously not been feasible. And this involves protein probes or FRET-based systems or luminescence-based systems. And one good example of this is we have an ongoing collaboration with our friends at Promega, where we've been able to show we can deliver their large bit system. So this is a system that, if you could deliver it into a cell, will tag a protein of interest and generate a luminescent signal. And we were showing that in live cells, we can actually use this to get a strong luminescent signal for the protein of interest. And then if you added a degrader to this, you could watch it degrade away, therefore giving a much more realistic view of kinetics within live cells, as opposed to the much more artificial systems of biochemical assays or lysates. Finally, the larger scale of what we've been implementing at is for clinical use. And in this case, we have these cartridges that are compatible with many different systems. We have a number of our partners listed here. Some of the larger ones like Thermo Fisher and Fresenius Kabi have a lot of equipment out in the field. Some of our more emerging partners like Multiply Labs and Limula have exciting new technologies that are being implemented for automation and simplification of cell therapy manufacturing. And to give you a flavor for how we think about this area is we're just a cartridge that tube welds onto existing kits and allows you to do delivery at scale. For example, in this case, you're looking at delivery to PBMCs, where we just connected one of our cartridges to Thermo's Rotea and showed that we could get robust delivery and viability across over 1 billion cells. And one further illustration has been with our partners at Fresenius Kabi and ScaleReady, where we showed that if you take T cells and deliver to them at small scale to do an edit, and then expand them in a G-Rex, and following that expansion, take those cells and put them through a portal cartridge at clinical scale, in the end of the day, you're going to see that you had pretty good normal expansion, maintained viability throughout this process, have a pretty high knockout efficiency, and ultimately get strong RNA expression on the other side as well. And from our past experience at Squeeze, where we were doing mechanical delivery in the clinic, we had also shown that across patients, these processes can be quite consistent. So this is some of our old data that was published a few years ago, where what you can see on the right is that we can actually get pretty consistent delivery and viability across these cells because mechanical delivery ultimately appears to be consistent patient to patient, where each of those dots is a different donor sample, and we can keep it pretty tight on viability, delivery, and recovery. And because of all these advantages, one of the partnerships we've been particularly excited about is our work with Fresenius Kabi, where they are generating a point-of-care machine where we're getting integrated as the key delivery system, while they also have other subsystems for cell separation and washing and formulation, et cetera, such that you can collectively have a machine that can just do the whole cell therapy process in a compact hospital-based or even bedside format, which we think will really enable a much broader use case for these cell therapies. Now, looking beyond our device side use cases and everything we enable there, another space that we work a lot with partners on and are excited about for the longer term is how to program cells in new ways. So if you think about most of what people currently do, looking at the left of this slide, most of the time people are programming cell therapies using a genetic modification. So they use a virus or a CRISPR complex to insert DNA or delete DNA. Whereas we think most of the future should be non-genetic modifications to cells. So delivery of things like mRNA, siRNA, circular RNA that can collectively modify multiple functions within a cell and give you the desired phenotype without the risks involved with doing a genetic modification. And our ultimate goal is, can we create a series of off-the-shelf software modules for cells, which allow you to then go and engineer different functions within cells simply by using mixtures of mRNA, siRNA, et cetera, and have these go after some of the big challenges today, which are not necessarily target specific. So 3 of the areas we really care about is 1, can you eliminate preconditioning for hematopoietic stem cells? Or can you enable better effector cell functions for T cells and NK cells by reprogramming them into a more memory-like state? And for iPSCs and regenerative medicine and more generally, can you leverage the delivery of transcription factors to push cells towards a particular cell fate of interest and create cell types that may not be accessible right now, or reprogram cells that wouldn't be accessible right now? And just to illustrate some of these as an example, we had published some work in the past showing that for hematopoietic stem cells, if you want to be able to get rid of preconditioning, one strategy you can potentially deploy is delivery of RNA for a mutant CXCR4, which allows you to home to the bone marrow niche, while a CXCR4 blocker like plerixafor might be mobilizing any of the endogenous HSCs. And you could even couple that with BCL2 overexpression, especially mutant forms of BCL2, such that you can use a BCL2 inhibitor to drive down the endogenous HSC population temporarily while your engineered cells are resistant to it and can have a competitive survival advantage when it comes to engrafting in the niche. And we think strategies like this might be able to take you to a world where you don't need to precondition patients and can have even a HSC stem cell therapy become an outpatient experience. The second example is thinking about improving T cell functions, where one of the strategies we had published on in the past is delivery of RNA for membrane-bound IL-2 and membrane-bound IL-12 that can collectively give the cells really powerful survival signals, memory phenotype signals, and proliferation signals. And some of the work we had shown from the past, in addition to improved tumor killing and persistence in vivo, was that you're able to shift more of these cells away from an effector memory or effector cell phenotype to a central memory phenotype, which can be ideal in principle for a lot of cell therapy applications where T cell phenotype has gotten in the way of efficacy. So with that, I wanted to thank everyone for joining, or those listening to this as a recording later. I would say the main takeaways for Portal are we're very flexible across the cell types people can engineer, the cargoes that we can put in, and the different scales that we can implement to that. And our priority has been enabling a lot of new things that weren't possible before. So for our beta program, we tend to discourage partnerships that are going to be much more incremental benefit, where it's going from 70% efficiency to 80% efficiency on something, but rather taking those big new leaps as far as novel assays for drug discovery or novel strategies for cell engineering, especially if it's non-genetic cell engineering. But our team is always happy to work with people thinking through what can be a useful implementation of Portal if it makes sense for our different partnerships out there. And with that, thanks a lot. Feel free to contact us and our team if you would like to chat, and hope everyone has a great holiday.